Welcome, everyone. We're going to start shortly. You can take a look at the screen. These are some expectations from you. In your calendar invitation, there were links to materials, including the introductory webinar that our leadership team recorded. This is going to be a little different webinar series. We had the overview recording that gives you background and introduction, and then we're going to get right into the 6 webinars. The overall theme is the landscape of early neuropsychological changes in Alzheimer's disease and Alzheimer's disease-related dementia. This first webinar is on the assessment of neuropsychological change across the lifespan. We have 3 wonderful speakers who will be speaking for 10 minutes, and then each of the talks will have 5 minutes of Q&A, and then at the end we'll have 5 minutes of closing remarks from our moderator, Lisa Barnes, and then 10 minutes of general discussion. Please use the chat for any questions. We'll try to get to as many as we can, but we will be recording this as well as the text in the chat box. If you have any links or anything else you think are important for us to record, please share in the chat as well. Without further ado, I will introduce the first speaker. On behalf of the leadership committee, myself, Luke Stokel at NIA in the Division of Behavioral and Social Research, Bob Bilder at UCLA, Maya Geddes at McGill, and Laura Germain at McLean Hospital and Harvard Medical School. We welcome Deanna Barch to kick us off. Thank you.
All right, thank you so much. Let me share my slides. So for those of you who don't know me, my name is Deanna Barch. I am faculty in psychological and brain sciences and psychiatry and radiology at Washington University in St. Louis. And I think that Luke asked me to speak in part because of my role in a number of initiatives in the field that have been looking at lifespan development and also the kind of optimization and translation of tasks from cognitive neuroscience for use in clinical populations, and in particular a consortium called the Cognitive Neuroscience Task Reliability and Clinical Applications Consortium. And although this work has focused on translation to people with serious mental illness, I think many of the same issues are relevant for translating, for example, from cognitive neuroscience to, being able to use tasks across the lifespan as well. So I want to talk a little bit about sort of lessons learned from this work and how it might be relevant to the goal of understanding sort of lifespan risk factors for aging, both healthy and disordered aging. So we know that kind of the tools and constructs of clinical neuropsychology, more well-established tasks, have many opportunities, right? They are often well-developed and very validated. They often have extensive norming data, and in a few cases across at least chunks of the lifespan that make them pretty usable. They're increasingly available on computerized platforms, although not always. And there have been attempts to measure across the lifespan. Some examples that I'm sure most are familiar with are the NIH Toolbox, which was spearheaded by the National Institute on Aging and really is geared towards measuring cognition from ages 3 to 80 plus years. There's also now efforts to move to mobile platforms, cell phone based assessments of cognition. That's the NIH Mobile Toolbox. And you can use these really interesting and often very large existing standardized databases to see if we can kind of get new or novel information out of these tasks that might be informative for understanding cognitive aging. But I think, there are also challenges associated with clinical neuropsychological tasks, right? They are often global measures of many different constructs, and that can be both good and bad. It's not to say bad if you're just after what's my biggest predictor, because sometimes messy tasks that measure many constructs might give you great prediction in some cases. But they also often don't make really clear contact with cognitive neuroscience literature or animal work. And that makes it sometimes hard to understand mechanisms or pathways that might be informative to treatment development. we often don't have a lot of information about psychometrics or construct validity across the lifespan. And there's, in the kind of more traditional clinical neuropsychology literature, there's very little focus on processes or mechanisms other than your more traditional cold cognitive tasks, working memory, episodic memory, language, processing speed. So not much focus on things like emotion processing or emotion recognition or reward or motivation or effort and apathy, social cognition. And those are all things that might be also very relevant to understanding the course of kind of cognitive aging and risk for dementia across the lifespan. So I think there's been a lot of use of, a lot of interest in sort of pulling tools and constructs from cognitive and affective neuroscience, where there's a lot of opportunities potentially to measure more specific deficits in kind of more, discrete cognitive systems. They're often linked to identifiable neural systems, and that can be helpful for mechanistic work, maybe functionally regulated by neuromodulatory systems that we can identify. And in some cases, let us try to distinguish between things like very specific deficits in particular domains versus more generalized deficits, which again can be really helpful for prediction and treatment development, and really letting us sort of bridge human and animal models of cognition. And I will say another thing about the kind of cognitive affective neuroscience domain is that there's much more work in that domain on constructs like reward and motivation and emotion and social function that we could pull into trying to understand kind of, aging across the lifespan. There are, of course, challenges, though, and the CNTRACs consortium has been trying to address these challenges, right? Like figuring out what are the best things that we should be measuring, what are the right constructs. There's often been really, frankly, very little psychometric work that's been done on kind of cognitive neuroscience tasks, and often some challenges about the practicality of administration, meaning they can be long and boring. And a lot of variability across investigators. We often don't know anything about test-retest reliability on many of these tasks. We don't know if there's a standard version that everybody agrees upon. And people may have many different tasks for measuring the same construct, right? So we need to think about sort of harmonizing parameters and manipulations and understanding whether the construct validity of the measurements derived from these tasks are the same or different across the lifespan. I think that's a challenge actually across both more traditional clinical neuropsychology and cognitive neuroscience, affective neuroscience tasks. Again, the issues with psychometrics. I'm going to give you one example of work that we've done in CNTRACs trying to kind of start to address these sorts of issues in one domain. So this is focusing on relational encoding and retrieval, which is sort of a specific form of memory encoding and retrieval that is thought to have specific neural substrates. And, we would want to be able to dissociate from other forms of memory retrieval. And I picked this example in part because it may be particularly relevant and salient to understanding both healthy and disordered aging. And so we focused on a task called the RISE, or relational and item-specific encoding and retrieval. And we picked this task because it had a very well characterized neural basis already. Our focus was on serious mental illness, so we knew there was preliminary evidence that it elicited impairments in people with schizophrenia. There's a lot of normative data already in both animals and humans. And we thought it was going to address some of these concerns about being able to distinguish between generalized and specific deficits. It could be used in an imaging paradigm, and it could be used in animal models. So we felt that those were all things that would, make translation and sort of animal modeling very useful. And so we did a lot of work with this task. It's pretty simple. You simply see pictures and you make one of two decisions about them. If you're focusing on just individual items, you simply ask, is this a living thing or not? Sort of deep semantic coding. And if we're focusing on relational encoding, we ask people to make a judgment that formally requires them to judge the relationships between two items. And in this case, it's whether one item could fit inside the other item. So really pushing for relational encoding, which is thought to pull on neural systems that might be dissociable from item-specific encoding. And then we have people do recognition tasks, either recognizing individual items, was it seen before or not, Or we have them do a relational recognition task where we ask, were these presented together previously or not? And when you do the relational recognition, all the items are old. They're just either correctly paired or incorrectly paired. So you're really focusing on people's memory for the relationships among things and not their memory for the individual items. Again, because that is thought to engage dissociable neural systems. And so after a number of studies, right, we were able to generate information about the psychometric and kind of practicalities of these sort of tasks, right? Showing that the various measures had reasonable internal consistency, good effect sizes for discriminating between folks with schizophrenia and healthy controls, and I would say good to modest test-retest reliability. You know, I think I could talk for an hour and a half about some of the challenges of doing this kind of translation, as important as it is, right? You know, it's challenging to do and to try to hone things down into very kind of practical forms, but I think it's well worth it when we think about kind of the benefits that we might be able to accrue from using these kinds of tasks that might be better linked to the neuroscience of what we know about memory and different forms of memory, and also very doable in a variety of kinds of animal models that might help us with translation.
So you have 1 minute, Deanna?
Okay, I will just say we've also been able to use this in imaging, and we get very nice results pulling out both kind of frontal and hippocampal systems. So I'll focus just on kind of open questions and next step. I think there's a lot of interest in the field now in using computational approaches to do an even better job of identifying dissociable mechanisms and processes. But there again, we know little about the psychometrics of computationally derived parameters. The current incarnation of CNTRACs is actually focusing on that. And I will say that, you know, these things, particularly the computational approaches, have not been well studied across the lifespan. We haven't done a great job of looking at kind of cultural and socioeconomic status effects and how those impact both construct validity and the psychometrics of things. And, you know, we need to continue thinking about implementation and dissemination. CNTRACs has worked a lot on making things kind of user-friendly and implementable in the field, and reducing burden by shortening things and making things publicly accessible, but there is much more work to be done here. And also trying to do a better job connecting these functions to people's everyday life. You know, in our own work, we've done a lot of like self-report and informant report and proxy measures of function, but we're also starting to add things like mobile sensing and ecological momentary assessment to get a better sense of how well these kinds of tasks in the lab predict what people are doing, experiencing, struggling with, thriving with in everyday life. So I think there's a lot of opportunities here, and I know some of our other speakers will address some of these opportunities as well. All right, I will stop there.
Thanks, Lisa. We're going to open it up to Q&A, and Lisa, you can run.
Yeah, great. Thanks, Deanna. That was really great presentation. The floor is now open for questions. You can put them in the chat. But Deanna, I will start off with the first question. I came from the cognitive neuroscience world before I was in epidemiology. And usually our samples were small samples of highly educated college students. So my question is, do we know how these tests perform in older adults? And do we have any information about how performance correlates with ADRD biomarkers?
So we haven't, in our own work, right? Like my work is focused only on mental illness. So I haven't done any work in AD, but others are starting to do work with these. But I will say in all of our development work, we have very much avoided college undergraduates and have focused on, you know, more representative community populations, which is really important to do when your target audience is serious mental illness, right? Because that is, you know, unfortunately often overrepresented in, you know, lower SES populations. I can tell you we have looked within our samples at age-related effects, and we do quite clearly see age-related effects, but we haven't personally looked at them in relationship to AD biomarkers.
Okay, great, thank you. There's a question in the chat from Chandra. how much longitudinal data is being sought for the RISE tasks?
So we have done longitudinal work over the course of shorter time periods, right? Like months, not years. But again, there are since all of our tasks are publicly available, there are people who are using them in longitudinal studies to look at changes over time. And we actually built them to be able to do that, as have many other people, by having multiple stimulus sets, so some forms of cognition, particularly in the domain of memory, right? You have to worry about practice effects and, having multiple stimulus sets. So we set it up so that there are multiple stimulus sets being able to use that. But someone put in the chat, right, that the toolbox, certainly that has been used longitudinally. It also has multiple stimulus sets for the tests that are needed. Although I think there's still more work that needs to be done there, because we don't have an infinite number of stimulus sets for a lot of these tasks. And if you're really, I think the toolbox might have like 2 versions of the memory tasks. And we don't know how much of a difference that makes, right? Like, do people really remember the stimuli over 2 years or not? I think to some extent you can view that as a feature, as not just a bug, because if you're looking at different kinds of populations and they're all getting that same repeated experience, you might actually see that sort of failure to improve as a function of repeat practice ends up being an interesting marker of potential cognitive challenge as well.
Great, thank you so much, Deanna, for that great presentation and the discussion. Let's move on to our next speaker so that we can stay on time. Chandra, do you want to share your screen? Great, thank you.
Thank you. I'm very pleased to be part of this webinar. I am going to be speaking about reliably measured declines, gains, and maintenance, reserved concepts, and signal and noise. I am a professor of psychology at University of California, Riverside, and I am a part of large multi-site projects that look at twin and family approaches to individual differences in cognitive functioning and aging and dementia outcomes. One aspect I think that has not been as much paid attention to in terms of individual difference characteristics is peak performance. We certainly have a lot of information on average from a number of studies, but I think we would be well positioned to address this in terms of time to criterion models. And in as much as we'd like to distinguish change, including both declines and gains, particularly for lifespan models, but also in terms of treatments and interventions, understanding peak performance and the individual differences across domains could be a very fundamental set of information to understand as people start to decline in one domain, but maintain skills in other domains, and where they may start to try to buffer losses and so forth. The model that's pictured here from an article from McNeish et al. Is suggesting that we can model a kind of trajectory up until a change offset occurs at some maximum or some minimum. And of course, with models like these, repeated and intensive longitudinal measurements are essential. I want to give a representation of applying this kind of model earlier in the lifespan with the view that we could apply such models at various points in the lifespan and in terms of intervention models as well. This is a structured latent growth curve model of cognitive change. It's a Gompertz model, and there are 3 features of interest that are allowed to show individual differences. And the first is A, which is the total change from a lower to an upper asymptote, R, which is the rate of change of approach to the asymptote, and D, which is the age at which accelerated change is observed. And this is a representation of data from the Colorado Adoption Project, where we were looking at adoption status as a marker of early vulnerability to stress and looking at their performance on the Colorado Perceptual Speed Task between ages 8 to the early 30s. And in this model, we were able to address a few interesting aspects in terms of individual differences. And that again is this total change from lower to upper asymptote. So 27 points gained among non-adoptees, but a few points less for adoptees. We can look at the rate of increasing approach to the asymptote, and then the age at which accelerated change occurs. And most fundamentally, in terms of individual differences, we can look at the degree to which these various aspects of the growth features vary across individuals and target them for prediction. Other aspects of measurement earlier in the lifespan that are salient and still remain to be fully understood is the extent to which childhood and adolescent represent a period of salient early life reserves that may in some ways have some salience and benefit even beyond later gains in reserves. So we need further replications and diverse samples and cohort comparisons and prediction beyond, for instance, genetic factors such as ApoE epsilon 4 carriers. There are relatively few studies with multiple assessments in childhood and few studies combining imaging and cognitive that are genetically informative in this way. And we would be well positioned to try and leverage additional resources into extant studies as well as new studies. And just to give a frame of reference for the particular early salience of early life reserves is an example from the VETSA study where higher young adult general cognitive ability and stronger maintenance of young adult general cognitive ability were related to higher levels of specific cognitive abilities between ages 51 and 73. But most particularly salient to my point is that after adjusting for current general cognitive ability in midlife, higher young adult general cognitive ability was associated with better episodic memory, working memory, and executive function across midlives, suggesting that young adult general ability pays it forward into midlife and possibly beyond. And it would be important to test further replications in females and diverse samples and in across a variety of specific cognitive domains. Other aspects where I think we need to build some increased effort is understanding the contributions of genetic factors to AD risk and other related constructs across the lifespan. Here we have a twin model of Alzheimer's disease, plus including polygenic risk scores. And the red part of the bar indicates how much is left to be understood, even when including polygenic scores and the ApoE region. And so even if we consider other studies of SNP heritability of individuals, while they may show some greater contribution from these measured genetic variants and polygenic factors, there still is a vast amount of the risk that remains to be understood. And likewise for genetic biomarkers or indices of gene-environment interplay, epigenetic work suggests that there may be some stronger heritability of DNA methylation for aging-related gene pathways, which may reflect gene-environment interplay and adaptations to aging. However, much of what we know is based on very small samples that have DNA methylation measures, and especially so for longitudinal studies. We also understand that environmental variants may increase with age, but what I think is still needed is a lifespan perspective of DNA methylation in intensively longitudinally measured samples. So with that, I think we need to be investing in new and diverse samples with intensive longitudinal measurements and at earlier periods of the lifespan, with investments in new GWAS of not only AD risk, but building on some recent work on AD resilience, as well as methylation and other biomarkers of relevance across development. We also need to consider reinvestments into GWAS of even more typically measured samples in European ancestry, because we keep—
One more minute, Chandra, one more minute.
Okay, we keep building upon studies without sort of refreshing cohorts. But we also need to, of course, leverage additional resources into understanding genetic pathways, for instance, in understudied populations. And so, for example, a recent GWAS of African-American individuals suggests that while some of the same genetic pathways shown here on the left-hand side of the slide may be similar, there may be different genes participating, and there may be other gene pathways, such as kidney development, that may be salient. So we need better understanding of this across diverse samples. We also need to invest in studies of reference samples for biomarkers, and here is shown for neurofilament light, which is one useful example of trying to understand cut points of interest. But more studies are needed for other biomarkers of salience for ADRD risk. I don't have much time to talk about this, but a promising approach is to also consider lack of coherence across a battery of tests or within a task, but we don't have a fundamental understanding of reliability of dispersion measures, maybe by design, as they reflect a lack of consistency. But we also need to understand genetic and environmental contributors and longitudinal approaches to understand the emergence of dispersion and variability within and across tasks. So with that, I'll just end with a suggestion that we build upon existing genetically informative studies with more sampling for biomarkers, but also investing in new and diverse cohorts within extant studies where possible. Thank you.
Great, thank you so much. That was really interesting and exciting. I'll open it up now for questions. I do see some things in the chat related to Deanna's talk, and we'll do that later. So here's a question from Rob, sorry. Chandra's point about dispersion. It might help to use item response theory and person fit statistics to supplement measures like dispersion that focus on total scores. Any comment to that, Chandra?
I think that's a great point. I mean, I think we have to kind of step away from more traditional measurement models to understand this. And I think that is certainly a viable approach to understanding it, and possibly also using other prediction models as well.
Okay, let's see, John King, there'll be NFL data and intensive cognitive testing in 2,900 people over 65 in HRS, okay? And Luke, Bob, sorry. Can you please elaborate on your point that you raised about dispersion, please?
Yes, so measures of dispersion would include the extent to which people perform sort of consistently across a set of domains or not, and that these kinds of measures across a battery of tests or within a task, so the lack of consistency within multiple trials of a task, are predictive of memory and cognitive dysfunction and dementia, but have been primarily looked at in older samples rather than in younger samples. And so that's, I hope anyway, that gives a little bit of a picture of this type of work. I think in our own applications in twin and family studies, we don't see a lot of sibling similarity for these kinds of measures as we might expect. And so perhaps leveraging some of the approaches discussed in the chat might be helpful to try to understand this a bit better.
Okay, one more question for you from Deanna. how frequently does the same study directly pit peak performance versus average performance versus within task or across task dispersion to outcomes? And if so, what tends to be most predictive?
That's a great question. I don't think the same set of studies have pitted all these things against each other. So I think that that would be a very interesting aspect to look at. I mean, certainly average performance, may correlate with dispersion, but in terms of pitting peak performance, I haven't seen much work on that. I don't know if others in this group have.
Okay, let's go on. Thank you very much, Chandra. I'm sorry, there's so much in the chat. Let us go on to Yakil next, please. We can come back to some of these points in the general discussion.
So thank you, everybody. Let me just close here. Good morning. So my name is Yakil Quiroz. I'm an associate professor at the Harvard Medical School. And today I'm going to be presenting on some of the work that we are doing here in the lab about improving early detection for cognitive decline. And this is a little bit different from what it has been presented before, but thank you for the opportunity to share this work. So as you may know, so Alzheimer's disease is now conceptualized as a continuum, and we are talking about the clinical Alzheimer's disease, which is a phase that goes before mild cognitive impairment and dementia. And we know that most of the treatments that are now available are symptomatic treatments, and the goal of understanding preclinical Alzheimer's disease is to be able to delay the clinical onset and in the future to actually prevent the decline of cognition. And this is important because preclinical detection of Alzheimer's disease can actually allow us for the early initiation of treatments and also for the prevention of the disease, as I mentioned. So now we are talking about prevention trials, and this is important because the individuals that are actually selecting for prevention trials are selected because they don't have clinical symptoms or they don't have cognitive impairment in the standardized instruments, and they also selected based on biomarker evidence that makes them at risk for progressing to Alzheimer's disease, dementia in particular. So what we are actually learning is that the measures that we have available, they are not the best measures to actually detect some of the subtle cognitive changes that the individuals in clinical normal individuals may have, and they are not the best measures to actually help us to track changes or even like to be able to see what individuals are going to progress into dementia. So there is a big need for modify or improve the measures that we have. So in my lab, one of the goals that we have is to actually improve some of those measures that can actually help us to detect some of the subtle changes in individuals that are cognitively unimpaired, studying individuals that are at increased risk for Alzheimer's disease. The way that we actually have been doing it is understanding individuals from a Colombian family that have genetic risk for a autosomal dominant Alzheimer's disease due to the mutation that is the presenting one E288 mutation. Individuals from these families, they actually develop mild cognitive impairment at the age of 44 and dementia at the age of 49. And we know that because they carry this gene, they actually will develop dementia with near 100% certainty. And they also have a biomarker and other clinical symptomatology that are very similar to sporadic Alzheimer's disease. Our work has actually also told us that they have a amyloid pathology as early as 28 years old, and they also have increased tau pathology in their late 30s. So which pretty much means that they actually have pathology in their brain several years before they have clinical symptoms. So we are using this opportunity to work with these individuals to characterize their cognition, particularly in the preclinical stages, going very early in the disease, to see what are the earliest changes in cognition that can help us to predict and understand how their cognition is going to decline as they get closer to the clinical onset or the dementia. So some of the work that we have been doing, the first work that we actually did with them was actually taking some of the measures that have shown some evidence in the clinical Alzheimer's disease that was based on some of the findings from the Neuroscience. So the first one was this one, that it was the Face-Name Associative Memory Exam that was a work done with Dr. Risa Sperling and Doreen Renz. We actually validated and adapted these measures for the Spanish, Latin American Spanish. And that was the first work, going back to what was mentioned before in terms of getting validated, culturally validated norms. We actually developed norms for these tests in Spanish. And after that, we were able to use this measure with the Colombian Kindred, that is the second paper that we have, to see if the performance in the test was associated or not with pathology and biomarkers. So when the first thing that we have here is just the behavioral performance was actually able to distinguish carriers from non-carriers, and this is cognitively intact carriers, meaning like not even with cognitive concerns, between carriers and non-carriers. So meaning like the carriers, the ones that were pretty much at risk to develop dementia, they already have lower performance, even within the normal range, compared to the non-carriers. And then we've actually compared the performance with the pathology. Here we have the performance and the amyloid. You can see that those that have lower performance in the Face-Name, they also have greater burden for amyloid pathology. And here you can see that the lower performance in the task was also associated with greater burden on tau pathology. When we look at their brains and see where in the brain exactly the performance was associated with this pathology, you can see with either amyloid and tau, the regions of the brains that were actually not associated, but actually the regions that were highly associated with Alzheimer's disease, including the middle temporal lobe regions, the precuneus, and in the amyloid in particular, it was widespread. So pretty much showing us that individuals that have no symptoms, there are several, in this case, decades before showing clinical symptoms or even meeting criteria for mild cognitive impairment, they are already showing these cognitive changes in this test. So we did something similar. It's another measure that also has some evidence from the neuroscience, that is the color shade binding test that Dr. Mario Parra has used before. And we showed something similar, meaning like performance in the task was also able to distinguish carriers from non-carriers, and it was also associated with amyloid pathology and tau pathology. Then we took it to the next level and we started designing our own task. And we have actually designed 3 different tasks. This is the first one that is now like the MESERO 2.0. We have designed like different versions. And in this one, we actually decided with the idea of being able to put it also in the fMRI, so to be able to understand the brain regions that were supporting this kind of processing, and it's the semantically related objects. So individuals learn either 4 or 6 semantically related or unrelated objects. And you can see the performance in this task was also able to distinguish carriers from non-carriers and design like even performance, just behavioral performance within the normal range. And as you can see here, even very close, the carriers were already lower than compared to the non-carriers. In the last couple of minutes, I wanted to mention some of the ongoing work, some of the work that is unpublished, but I'm very excited and take this kind of like novel approaches that we are exploring in the lab. The first one is what we call MAPP remote. And this is like everything that we are actually learning with these measures. We created an online platform that is actually called madremote.com, that we're validating these measures with older individuals, including individuals with subjective cognitive decline, individuals with MCI and AD patients, and in Boston with individuals that are enrolled in the Boston Latino Aging Study. In the first phase, we are doing the supervised computerized versions of this task. And in the second phase, we are actually doing validating the task using the smartphones. The second slide that I want to highlight is also like a novel approach that we're trying to explore. Like as part of the pilot slide that we are developing, we are pairing in a sleep study that we are doing to understand long-term forgetting. And the same, like individuals coming to the clinic for an overnight sleep to do the sleep study. So the first day they go through memory testing, that is what it shows here. They spend the night in the clinic. So the next day they go through a second memory testing. And 1 week later, they're actually already, they go to testing number 3 to test the recall delayed recall of what they learned the first day. And as you may imagine, the first day, these are cognitively intact individuals. So the carriers and the non-carriers in the day one, they are exactly the same. But preliminary results actually suggest that the longer recall intervals may be more sensitive to detect subtle changes. We are already seeing difference in day 2. And of course, by day 3, carriers are performing worse than the non-carriers. The third study that I want to mention is what we call semantic fluency clustering. This is something that it has been studying before. It's a qualitative approach that is a typical 1 minute individuals are asked to say as many animals as they can recall. And then we actually go and take a look at how many animals are actually named in clusters. And here you can see that there is no difference in how many animals were named in clusters between carriers and non-carriers. We actually look in terms of how the associations were with pathology. We can see that the carriers actually have a stronger association between the number of animals that were in cluster compared to the non-carriers. And this was associated with pathology in terms of amyloid and tau. Even more, when you correct for all the comparisons.
1 minute, Yakil.
Yeah, normal one. You can see that the pathology was exactly the regions that you expected, and in particular with tau, especially in the entorhinal cortex. To conclude, traditional neuropsych measures may be optimal for the early detection of preclinical stages. We need to improve the cognitive measures that we have, as we want to be able to detect changes in preclinical individuals, preclinical stages. We need novel approaches, including qualitative approaches, novel digital technologies, and there is a critical need to develop and use more culturally and linguistically valid measures. Thank you very much for your attention.
Thank you, Yakil. That was wonderful. Let's open it up for questions to Yakil. Yakil, I will start with a question for you. Actually, I have 2 questions. So in your population with the rare mutations, do you know how the accumulation of amyloid and tau over time relates to performance on those tests? And the second question is, in that one graph you showed, there was a lot of variability in the association between performance and pathology in the carriers. Do you think that's a function of the sample size, or saying something about maybe resilience or ability to tolerate pathology in the carriers?
So the answer to the first one is like, we haven't actually looked at the longitudinal data. That's another step. So we are collecting longitudinal data, but we haven't actually looked at that. So that's like a future direction. It will be interesting to see how the accumulation over time relates to performance. We haven't done it. And the second one is just, I think there's as a matter of the sample size. The sample size that I was showing, especially the ongoing work, is no. So the work that I presented, the sample size is relatively small.
Okay, great, thank you. There's a question from Ganesh. For the semantic task, are you examining cluster size, number, and switches between them? There's been some great work in stroke subacute populations.
Thanks for the question, Ganesh. Yes, in this paper, we actually look at the cluster size and also the switching and the number of items that were included in the clusters. And the one that was most sensitive was the number of items that were in the clusters.
Great. Any other questions for Yakil? Okay, if not, I will offer some final words before we open it up to general discussion. Is that okay, Luke?
Yep, go for it.
Great. Okay, thank you to all of the speakers for this really informative webinar. And I'd like to also thank the webinar leadership team and NIA for hosting the series. As some of you know, I am following a cohort of older African Americans who we recruit from the community and enroll without dementia. And we give them annual assessments of cognitive function to measure change over time. But because participants enroll without dementia, we're able to observe changes in cognitive function before the clinical symptoms of Alzheimer's appear. However, there's a couple of challenges, at least 2, with this design. First, you have to be at least 65 years or older to enroll. So we're missing people who don't make it to 65, and changes that may have occurred before the age of 65 are not captured. So the level of cognition that we observe at enrollment is assumed to be their starting level, when in fact they might have already started a decline towards disease. The second challenge is that these tests themselves are often insensitive to subtle changes, and in many cases culturally biased against a population that has experienced disadvantage in educational experience, equality, and other structural inequities that we know affect cognitive test performance. So this webinar topic is exciting and opens the door to innovative ways to advance our understanding of cognition and other functions in our more vulnerable populations. So I'd like to offer just 3 points that I think we should think about as research priorities in this space. So one of the challenges that's been identified is health disparities and equity issues. And the leadership team had a guiding principle that the test should be inclusive, considering individual differences in language, culture, and identities. I really like the cognitive neuroscience tests and techniques that were introduced today, especially the prospect of measuring, other important components like reward, motivation, and emotion, and things like that. But as acknowledged, one key barrier is the lack of data in underrepresented populations on these metrics. Like most clinical research, samples tend to be homogeneous and white, and race, ethnicity, and culture are usually not considered in analyses, except for in Yakil's work, of course. So we have no idea how our underrepresented groups will perform on these tests. And if they are as correlated with education as traditional tests, could their use make the disparities even larger? Before we adopt any new measures, it will be important to include the populations who we think are most at risk. And ultimately, studies to design and validate new instruments should be conducted in representative samples from the population and powered for validation across race and ethnic groups. The second point that's related to the subject of inclusiveness is the incorporation of social determinants of health. We know that sources of underperformance on traditional neuropsych tests in these minoritized populations are influenced by lower educational attainment and quality, lack of familiarity with test-taking procedures or items on the test, and other structural inequities. For example, a number of studies have demonstrated that culturally relevant factors like acculturation, experiences of discrimination, and residential segregation influence performance on the traditional tests. Whether or how such variables might affect some cognitive neuroscience approaches, for example, is unknown because we haven't examined these constructs in these types of studies. Also, what's the role of comorbid health conditions that we know are more prevalent in minoritized populations, like hypertension and diabetes? All of these cultural constructs and health conditions introduce heterogeneity and would need to be modeled if we believe that aging trajectories are influenced by contextual factors that increase decline for some people compared to others. Finally, I think it'll be important to design future studies that have more ecological validity and measure cognition within the context of real life, outside the artificial settings of a laboratory or a clinic. In my longitudinal study, we've been able to employ passive sensing techniques in the home to measure room transitions as a potentially sensitive marker of everyday function. And we recently published a study where we found that indoor room transitions measured over a month were associated with MCI, such that people with MCI had a different spatial temporal pattern of movement compared to those without MCI, suggesting that this might be a viable approach to detect gradual changes in daily activities in those at risk for dementia. Importantly, things like this is a culture and burden-free way to measure everyday function and can be coupled with other everyday activities like taking medications, driving, or moving through different activity spaces in the real world. So I'll end there. I thank you all for the opportunity to serve as moderator and add to this conversation. And let's now open it up for general discussion. So there were a few points that I wanted to come back to that were in the chat that I think are important for us to talk about. One of them was from Duke Han about the idea behind dispersion. He thinks it's a good one, and that there are probably other ways of looking at performance on a particular test other than what has been historically considered. And I think this is a really important point when we think about our underrepresented populations, where we know they're performing more poorly if you just look at total scores. So can we have a discussion about whether a metric like dispersion or variability, could those things be leveraged to help us understand our most vulnerable populations? Anybody want to take a crack at that? Speakers or others? Do you want to?
Yeah, I'll just jump in. I thought it was a really great idea that Chandra mentioned, and I'm trying to think more outside of the box. I think, we always sort of default to a total score or sort of even cut points and things like that we've used historically. But I think given the challenges that I think this group is trying to tackle, I think looking outside of the box at things like dispersion or variability or ways or approaches, from a qualitative almost standpoint to get at other metrics might be a way forward. I know there are a number of people that have started looking at item response, theory and other sorts of approaches. So I just think I'm a fan of those outside of the box, approaches to try to see how performance might be characterized in different ways. And whether or not that actually proves fruitful is another story, but I just really appreciated that point.
Great, thank you. Deanna wants to have a little fight with me.
I don't know.
I'm kidding.
I want to, that and, right? It's just, I think it's just not enough for us to say in this domain that we have a representative population, because if we're really trying to do individual level prediction, we absolutely have to know that that prediction holds within that subpopulation, and we can't get there with just sort of an average representation. And so we need to oversample, and it sounds like NIA is already doing that, right? But
Yes.
yeah.
No, great point. I think we're on the same page with that.
Right.
I mean, obviously there's challenges to doing that, the oversampling, but I think it is critical to move the field forward. And actually Duke had another point in here about the implications of not doing it, right? So he says, is it worth discussing further how not doing this, what we're talking about now, historically has impacted the field and might impact things like AI approaches? Anybody want to jump at that?
Actually, I just wanted to say something briefly about the previous topic, which people should be aware of. At NIA these days, if you're submitting an over-the-cap application that's about Alzheimer's disease and related dementias and therefore requires our approval, we were explicitly requiring people to actually say something about the diversity of the populations, the representativeness of those populations, at the outset, and it's actually one of the criteria that's been used in some cases to actually ask researchers to go back and perhaps think more strongly about that. Change is not happening overnight there, but I mean, it's a situation where, we're very, very conscious of the need to do this. There is no probably one size fits all answer, however, right? So you can go an HRS approach where you really are trying for a truly weightable population representative sort of thing. And it's wonderful when you can do it. It's difficult to get every single potential subgroup weighted up to the level you're going to need it in a sampling frame like that. There are definitely room for other sorts of studies, either more community studies or other sorts of things where you're focusing on, the disparities and discrepancies that you're really interested in looking at, rather than attempting to get one complete nationwide sample that sort of captures all the variability you need. And sorry, I stuck that in, and you probably need to remind people of the previous question you asked, Lisa, but I just wanted to get that out there because we are definitely, it's affecting our behavior there.
John, just a quick follow-up question to your first point. So it's, we had NIA or NIH had something like that with having us put a recruitment plan in for all of our studies. And I always found when I was on study section, that there wasn't a lot of consequences or follow-up. How is this going to be different?
So so that's another point too, is that there's another system that many of you who've run, actually, as of right now, it's been primarily clinical trials on ADRD, but it's going to expand beyond that. There's a new CRUMS system, CROMS, that is actually sort of helping us record in real time as people are accruing participants into trials, whether or not they're actually hitting the enrollment targets in a way that seems appropriate for the study and they're not going to fall way behind, things like this. So there's definitely a lot more attention and therefore reporting being made, requirements being made in some of these studies and more in the future. But you're right. I mean, at the end of days, it's a very tricky situation. A study section may be perfectly happy with a specific plan that we might wish to be stronger in some ways, and it's going to be definitely hard work to make those things happen. But you're right. I mean, in the past, and a recruitment plan might be, well, we'll recruit what we have in the community, full stop. And what's the scientific judgment to be made there? And that's always been difficult.
All right, thanks, John. I'd like to call on Laura or Jermaine to raise her very interesting question to the speakers. Go ahead, Laura.
Yeah, hi, my power came back on, so I'm no danger of my computer turning off anymore.
Yay.
So I think, as I was listening to the talks, there's this constant issue of how do we have all these different measures, and I think creating ad hoc measures makes a lot of sense if we want to get at specific constructs, especially constructs that might be really important for a certain population or a certain context. But then there's the issue of combining across data sets, right? And so we often end up with a lot of studies that can't be combined that are looking at very specific, interesting questions, and a bunch of studies that have, similar methods that can be combined, but then, you're either dealing with these factor analysis approaches where there's no mechanisms at all, like the G factor, or you're in a situation where everyone has to use the exact same measures that, might not be ideal for a certain population or a certain context. So I just love to have the speakers' thoughts on, things like computational modeling, dispersion, inter-individual variability. Like, do those provide us new ways to get mechanisms that can be combined in a way that allows for those differences between studies, but also allows us to bring them together in a really analytically meaningful way?
For the speakers. Go ahead,
Yeah.
Chandra.
No, because
Yes, I mean, to Laura's point, I think we can leverage dispersion and IIV measures across studies if, say, perhaps there was sufficient coverage across a set of cognitive domains and a sufficient number of tests. So at least there would be some kind of comparability in that sense. And then I think it would be possible to combine these kinds of statistics, even where the batteries are rather different. And then I would also suggest, and I think Marty has also commented on this, that using the person as their own control in longitudinal designs would also give us a understanding about whether variability measures or dispersion might be representing an interesting signal, beyond just cross-sectional kinds of work.
John?
Just super briefly, yeah, important point, Laura. I sent some stuff in the chat and we can talk about more of it later. There has been some recent work that I think is actually being potentially quite exciting with trying to establish under what conditions you can safely, as it were, sort of combine across cohorts. So one other interesting feature has been in the HRS family, the Harmonized Cognitive Assessment Protocol, or HCAP, exists for HRS in the US and English, but also for several cohorts around the world. And the question has been, so how do you contrast, how do you compare, how do you combine across those? And there's been some suggestive early good news about uses of some fairly sophisticated IRT-based approaches based on commonalities between items that may really get us a lot further than we would have thought, frankly, on some of these issues. I don't know if there's any way that we can do things with measurement batteries that are absolutely disjoint, but the amount of sharing that you need, the amount of common material may be actually less than we might have feared. So we'll see how that goes. As we're building some these very large data sets now, particularly with mobile tools and stuff, I think there's going to be a lot of work to try to get some of these things to work out. But the cool thing is there'll be enough data to work on to see if these things are feasible. Whereas to date, it's not always been clear.
Dana.
Excuse me, hi. I just wanted to let you know I did stick a reference in that may be of use or helpful that just came out from the Intensive Longitudinal Health Behavior Network, which is funded through OBSSR and partner ICs. And it does some of what you're talking about. Basically, the network has tried to leverage data and some of the things that they are capturing in different populations to answer questions separate from their original research ideas, and have harmonized, worked to harmonize all types of different data, GPS data, EMA data, etc. So if that's helpful, I hope so. It is in the chat.
Great, thank you. And Luke, we'll have access to the chat afterwards, right?
That's correct.
Okay, great.
And we have 1 more minute, Lisa.
Hey, 1 more minute.
Close out.
Yeah, so I don't know if there's any other burning questions or comments, but I just want to thank everyone, all the speakers and all of the attendees here today for this really great first webinar. I encourage everyone to go onto the box and look at the opening video, maybe look at the talks again and get some ideas for our meeting that's going to happen in April with everyone. So thank you all for tuning in today.
Thank you. Yeah, we'll have a recording of this video, and if you have other references to share, you can put them directly on Box or just send them to myself or Becky Rickless. I want to thank Becky as well for corralling us, and she does all the scheduling and all the work behind the scenes. So thanks a lot, Becky, and have a good day.
Bye-bye.